really trying to promote this culture, this new culture of open science, especially with the young scientists like many in the audience today. Normally I've done quite a few of these Scandinavian summer courses. Normally it means a nice trip in June to Sweden or Finland or Norway, but this time, okay, it's not like that. And I guess the positive is that we have many people from different countries, maybe more participants than normal. The other advantage is I don't have to deal with those mosquitoes. You have these giant mosquitoes in near the lakes in Sweden and Finland. And I remember being attacked by those. They're quite scary, I have to say. But okay, so that's maybe another positive of this, this kind of remote interactions. Anyways, let's talk today about NLRs, so plant immune receptors. But before I start that, I want to briefly introduce you to the sort of the direction my lab took in the last maybe 5 years, 5 to 10 years maybe, but it became better articulated in the last 5 years. We decided that we should focus our effort as much as we can on filling this gap between mechanistic research and evolutionary biology. And I think it's fair to say that in the past, these fields have been very segregated, and there is truly an opportunity there to bridge these two very critical parts of biology, of biological research. And really, especially in the post-genomics era, there's a lot of opportunities here to sort of combine these two approaches and hopefully generate new perspectives on how we do biological sciences. And this, really, this statement sums it up. This is kind of the motto of this field now. I think it's a budding field. You can see it with symbiosis, with other aspects of plant-microbe interaction. We can call it EVO and PMI if you like. Those on Twitter can follow hashtag EVO and PMI. So clearly, in the absence of an evolutionary perspective, our mechanistic understanding is unlikely to explain the messiness of biological systems. Let me briefly give you an example, which I think we'll go back to this at the end of the talk, and hopefully you can see the connection. Let's say you study wing function, and your model system are penguins. You're going to have a very warped view of what wings do, right? You will think that wings function in swimming, and you would be very confused by many features of wings, actually. The morphology, probably some of the mechanism, maybe even how the muscles are structured, all of that would be really difficult to interpret. You probably would publish very good papers and very interesting studies. But by studying that one single organism and not bringing in the evolutionary perspective or the comparative approach, you will have a really, I think you can agree, even if it's specifically correct interpretation of wing function, you will have a very narrow view of really what wings do. And you may even make some very silly statements like wings functioning as, you know, as flippers for swimming, etc., which are really fundamentally flawed if you take the bigger picture. So this is what, this is not my view. This is a view that's been around for a long time, except that the mechanistic people who do mechanistic research haven't really integrated that thinking until recently, I should say. And Francis Crick, for example, he eloquently wrote this in his book, that biologists must constantly keep in mind that what they study or what they see is not designed, but rather evolved. And he puzzled on the fact that, well, yeah, I mean, he thought that evolution really should play a much larger part in biological research. And this is far from the case, especially in molecular biology. Okay, so this is my kind of general perspective, and we'll get back to this at the end, and hopefully it generates some thoughts and ideas in your own minds. But if you want to read more about the topic, I encourage you to check this review I wrote called Can a Biologist Fix a Smartphone? And there is also a talk on YouTube on the same kind of topic. All right, so plant immune system. So I think you've been exposed to this already in this course, but one central dogma, if you like, in plant pathology is that most plant pathogens, or most plants are resistant to most plant pathogens, right? So disease is an exception, and what is maybe new in terms of our interpretation of this general observation that's been around for what, a century or something, is that the reason why plants are so good at fighting off most pathogens is because they have a very effective immune system. And that's really I think the concept that has crystallized over the last 20, 30 years, and I think now is really coming to its full fruition about the complexity and the capacity of the immune system to fight off so many potential pathogens and microbes and pests that are out there. And again, you've been exposed to this, so I'll go fairly quickly over it just to give you a little bit my perspective. Let's start from the pathogen side. We know that now pathogens, all kinds of pathogens, fungi, mycetes, bacteria, nematodes, aphids, and so on, secrete effectors. They modulate host processes in different compartments of the interface between the pathogen and the plant. But plants and plants do fight off. So plants have these immune receptors. Some of them are on the cell surface, some are intracellular, and these immune receptors can perceive these molecule coming from the pathogens, the effectors. So in that case, the effectors become liabilities, if you like, for the pathogen. They chip the wire and activate immunity. And we now know that this surveillance system, this immune surveillance system that plants have, is very effective at detecting perturbation caused by pathogens. More often than not, these are indirect perturbations that the plants have evolved to basically say, yes, I have a pathogen now. I'm facing a pathogen, let's activate immunity. So the immune receptors are obviously that first layer of the surveillance system, and they're extremely heavily studied. There are multiple classes, but the ones we'll talk about today are the NLRs, or these intracellular sensors, if you like, of invading pathogens. NLRs are these multi-domain proteins. They have a central domain, which is the MBR domain, which acts as a switch. This is how these proteins get switched on through conformational changes resulting from an ADP to ATP switch in this nucleotide binding domain. And there are additional domains in plants. The N-terminal domain is variable, but these proteins basically are sitting there in a plant cell in an inactive resting state. And when a pathogen effector comes that is detected, then they switch on into their active form. And this results in, more often than not, in the hypersensitive cell death response, which I'm sure you're familiar with. And this response, we can visualize it experimentally using this wonderful model plant, Benthamiana or Benthi, by doing these assays where we infiltrate with using agrobacterium, co-express an effector and a receptor. And when there is a matching interaction, we get the cell death response, and we have a positive for immunity, positive readout for immunity. The leaf here on the right, let me use the pen. That was better. The leaf here on the right shows a plant carrying a Benthamiana carrying a resistance gene, an NLR. In this case, you have resistance. In this case, you have susceptibility. So you can clearly see that there is also a cell death response associated frequently with pathogen resistance. Now, the first NLRs were identified back in the early 90s. This is a very heavily studied protein family, maybe the most heavily studied protein family, with over 450 experimentally validated NLRs. We had a lockdown project. We were stuck at home last year, and we decided, okay, let's go through the literature and catalog all the validated NLRs. So Yorgos Kourelis did this. This is now in bioRxiv and will be published hopefully soon. And this illustrates basically the diversity of NLRs that have been validated. 30 genera, more than 10 flowering plants, mostly from Arabidopsis, Solanaceae, and monocots cereal crops. This also illustrates the fact that we have all kinds of pathogens and pests being detected by NLRs. So essentially, pretty much every type of organism that can infect plants can be detected by NLR, including parasitic plants, incidentally. So there's one example of an NLR that function against the parasitic plants. There's probably more, but they haven't been studied yet. So I just wanted to highlight this. This is a really good reference article that I encourage you, if you're in this field, then this can be sort of a benchmark for what you're doing, and find out if you are finding new things, for example, and relate your work to previous literature. Okay, so cloned in the 90s, and for over 20 years, maybe 25 years, it was really struggled to understand how NLRs function. It took really completely new technology, the technology of biophysics, cryo-EM, getting the structure of activated oligomers to really figure out how NLRs function. And the reason is they don't function necessarily through your classical sort of MAP kinase pathways. They function through the formation of these oligomeric resistosomes, which then are activating the cell death in different ways, depending on the different classes of NLRs, but sometimes not necessarily through directly through activating a signaling pathway, although there's still some uncertainty in these models. But the point is that now we know that resistosomes can be formed by CC-type NLRs. So this is one class of NLRs that carry a coiled-coil domain at the N-terminus. And this is wonderful work out of Beijing, China, by Jiamin Zhu and Gigi Shai's labs. Really landmark discovery in our field. And they discovered that ZAR1, a CC-NLR of Arabidopsis, when activated, forms this oligomer, this pentamer, which has this wheel-type structure and is presumably inserting in the membrane and directly causing the hypersensitive response. More recently, we also have some two new resistosomes of the tier-type NLRs. How do these resistosomes cause cell death? Well, the mechanisms are very different depending on the type of NLR you have. So in the case of ZAR1, the CC-type NLR, the alpha helix, so the N-terminal alpha helix, which you can see here, which is normally buried in the structure of the inactive complex, flips out when this is activated, and then is protruding in the oligomer and is forming a pore-like structure that is inserting in the membrane, the plasma membrane, presumably. And this little molecular machine is acting as a channel where various components are going on. In particular, calcium has now been demonstrated to be going through this channel, but there could be additional molecules going through this pore. So we don't fully understand how the cell death takes place or how the immune response is activated, but this could be through perturbation of the membrane and could also be through this pore-like or channel-like activity. But for the rest of the talk, keep in mind this alpha helix. So this is the absolute N-terminus of ZAR1, which end up associating in this pentamer and forming this funnel-like structure in the inserted in the membrane and essentially critical to the activity of this resistosome. The tier-type NLRs function very differently. So these have this tier domain, which is essentially an enzyme domain at the N-terminus, and they function through hydrolysis of NAD. So they're essentially NADase, and by the formation of these various compounds, then this is activating through an unknown mechanism the immune response and the cell death response. So again, you can see already from the structures of these two very different classes of NLRs that their mode of action is completely different. And this is a theme that will be recurring in my talk, is essentially when we think, 20 years ago, people cloned NLRs, they all looked similar on the sequence level, they had similar domains. There was, okay, NLRs, one family, one function, etc. So remember the penguins, right? So these two different NLRs are functioning through completely unrelated mechanisms, if you like. They might be connected, but that's another part of the story we'll get to. Okay, so now let's see slides. Let's go back in time now. So that was basically the state of the art in our understanding of the molecular mechanisms of how NLRs function. But let's roll back the clock and go back a century ago to Roland Biffen, a very funny character who was working at the precursor of the John Innes Center. You can see him with his joke here about breeding some very special wheat. He basically was part of those scientists who rediscovered Mendel's laws of genetics, and he did that by studying rust diseases, and he discovered basically R genes. He was the first one, I suppose the first one, I'm not a historian, but people say he was the first one to basically show that there were single genes that were segregating, that were conferring resistance to a pathogen, in his case, to yellow rust pathogen. So that was early 20th century. 50 years later, Harold Floor came in and he brought this really very influential and very powerful model of the gene for gene resistance system. And he brought the pathogen perspective into this, right? So he brought the fact that pathogens have AVRs, what we call effectors today. And if a pathogen carry a particular version of this gene, the AVR effector, let's call it, and that's matching a resistance gene on the plant side, then you get this gene for gene match or this gene for gene interaction, then you get the resistance system. You need both components, both genes to be present on both sides to get the resistance and the activation of the immune response. Now, that was great. Floor's model is absolutely fantastic and has been extremely influential, in particular in plant breeding, for example, and has also guided the research because that really led to the discovery of these R genes and these AVR genes through genetic approaches. But now we're in the post-genomics era, so we need to think beyond classical genetics. We need to think in terms of systems, because we all know that this idea that there's a gene for this, there's a gene for cancer, there's a gene for resistance, there's a gene for virulence. This is a simplification. This is what geneticists use, basically based on variation in populations or in the germplasm they are working with. And that's basically essentially more or less a tool that allows you to identify those genes through classical genetics. The reality is, of course, is that all these traits have a much more complex genetic architecture that underpins them. It's obviously more than one gene that is involved. And the problem with classical genetics approaching this issue of genetic complexity or multi-genes and complex genetic architectures is it's difficult to tackle with classical genetics. You have issues like redundancy, epistasis, also sometimes phenotyping, quantitative traits. These are really issues that are very common in biology, but they complicate genetic analysis. So what geneticists do is they simplify the system and ignore essentially all these other genes and focus on the one gene that is really variable in the population they study. This has been really fantastic and great, but perhaps it's time to move beyond that and start thinking about our interactions, not simply in these gene for gene interaction systems, but start thinking in terms of complex systems where if you think of the effectors in red and the plant components in blue, then the reality is probably more like this than the oversimplified gene for gene model. So what is the biochemical translation of what I'm telling you about here? So when the first R genes and the first AVRs were discovered, people assumed immediately that it's a gene for gene interaction. In the biochemical sense, the easiest thing is we have a ligand and a receptor. So you have a ligand that binds a receptor, activates receptor, and then that results in a response to your HR, your immune system. This actually model, this biochemical sort of version of the gene for gene model has stayed around for quite a while. But now we know that the reality is not like that. This is the reality. The reality is that we have a very complex immune networks. We have indirect detection of pathogen effectors through GARDs or decoys, and even more complex versions of these. We have also cases where the receptors themselves, this is a very important point I will make, and I will get back to, the receptors themselves have diversified to start forming receptor networks. And these receptors start specializing either in sensing the pathogen or in executing the immune response. So essentially have a subfunctionalization of the receptor into this very specialized activities. And then, of course, we have the downstream components, which are also very variable and that can involve also different responses depending on the cell that is being sort of attacked or based, whether the cell is actually the one that is infected or neighboring cell, etc. These are concepts we're just starting to really scratch the surface of. So the point is really is that we have obviously every signaling pathway is a network. But what's really exciting here in our NLR and immune receptor biology is now we are reached a point that we realize that pretty much all of these receptors form networks. They form receptor networks. The receptors themselves are actually interacting with each other to underpin immunity in this very complicated fashion. So to sort of illustrate this a little bit more specifically, what we have with NLRs is we have this so-called sensor NLRs and the helper or executor NLRs. So these are the two major classes now of NLRs. It's just to simplify, it's more complex than that, but again, for simplicity, we have cases where we have one-to-one relationship where there's one sensor interacting with one executor, one helper, and there are cases here that are quite present in the literature that you can look up, like in Arabidopsis and rice. And then this goes all the way to networks where you have many sensors interacting with many helpers. And that's actually the NRC network that we discovered that I will tell you a little bit more about. To sort of give you the punchline, the work we've been doing, and this is something I'll get back to again a little bit later, is that the evolutionary model is that we started with the multifunctional receptor. So the ancestral state of these receptors, they can do both jobs. They can sense the pathogen, and they can execute the cell death. And then throughout evolutionary time, these multifunctional ancestral receptors have subfunctionalized into specialized sensors or into helpers. And in some cases, this is a theme that I don't know if this will come again in this course, but it's something we would work on that I won't have time to talk about. In some cases, the sensors have acquired actually new domains that are functioning as baits or decoys for detecting the effector. This is an interesting bit because these sensors have specialized so much that they actually are really evolving away from the classical architecture of an NLR and acquiring new domain, new extra domains that are specialized in sensing the pathogen. And those type of sensors rely on helpers to execute the cell death. Okay, so evolutionary model, multifunctional ancestral singleton, functionally singleton receptors into pairs and networks of specialized NLR proteins. Okay, so now let me introduce you to the network we work on. So this is now a project that's what, 5 years, 6 year old in my lab. It's I'll tell you a little bit how we stumbled onto this. But this is essentially how the network looks like now. It's getting too complicated to actually illustrate on a slide. But basically, all these guys here, these sensors, they are mostly classical R genes. So we have Rx, the famous R gene against potato virus X. We have BS2 against Xanthomonas. We have a number of Phytophthora R genes, RPI-BLB2 also, R1, PRF involved in bacterial resistance, AVR-PTO. So these are all classical R genes that were cloned over the years by various people through classical genetics. These are the ones that vary in the population that can also be introgressed to bring in resistance, like you can introduce RPI-BLB2 into potato, and you get very specific resistance to Phytophthora infestans carrying AVR-BLB2. What we discovered, what we stumbled upon is that all of these guys in this NRC network require these NRC helper NLRs. And these are also NLRs, these NRCs. For simplicity here, I'm showing you NRC 2, 3, and 4. Again, the system is actually more complex than that, but for simplicity, I'm showing you these major NRCs. And as you can see here, there is redundancy in the system. Like RPI-BLB2, for example, only requires NRC4. Doesn't work with the other NRCs, but if you take Rx, then you have NRC2, NRC3, and NRC4 equally redundantly working with Rx. This also explains, this redundancy explains why it's been tricky to identify these components, because you can't really, if you have 3 genes basically redundantly functioning in a response, it's going to be really hard to tackle that using classical genetics or even using mutagenesis. The reason we started working on this is really purely accidental. This is something I really love because you're sort of following the science here through my career, and then we don't know where things will take you. So years ago, we started working on cloning the R gene, the AVR effector matching the RPI BLB genes. These are from Solanum bilbocastanum. They were of commercial interest at the time because BASF wanted to commercialize the transgenic potato with these two R genes. And so in particular, we cloned these two guys using functional screens. That was the early days of effector biology. And we identified this AVR BLB2, this gene family in Phytophthora infestans that activates the R gene RPI BLB2. So Shihan Wu was a PhD student at the time. He joined the lab and he was interested in studying this AVR BLB2 RPI BLB2 association. And then he then identified an RC4. But you can see that there is a bit of luck here associated with this project because an RC4 is not redundant. So RPI BLB2 only works with an RC4. So if we didn't, if we had a redundant case, if we were working on RX, for example, or PRF, we probably wouldn't have identified the downstream component, right? So then, one thing led to another, and Shihan, through some really powerful comparative genomics and evolutionary analysis, identified that there is indeed a network here. The reason he could identify and predict that there is a network is he noticed that there is a phylogenetic structure to these interactions. So what you can see here is this is a phylogeny of NLRs from a whole bunch of different plant species, mostly asteroids. And what you can see is that the NRCs form a very nice clade here. So these are the helpers. And then what's really remarkable is all the NRC-dependent NLRs, all those R gene sensors, all of them fall into this big expanded clade, which is a sister clade to the NRC clade. So this is actually very well supported branches, both this one, which suggests multifunctional ancestor, and then this clade here of the sensors, the R genes, and then this clade here of the helpers. So it does look like this, basically this network, which is massively expanded in Solanaceae. I mean, some of the species, 50% of the NLRs belong to this clade, this NRC superclade. This network has evolved probably from a singleton that duplicated, maybe into a sensor helper, and then went on to expand in asteroid species and Solanaceae in particular into this very massive network. So that was really exciting, and we could sort of estimate that the expansion, at least, of the network occurred about 100 million years ago, before most asteroid species really split from each other. And then again, it's a massive expansion. And as you can expect, the R genes, the one that evolved to detect bacteria, viruses, effectors, aphids, nematodes, what have you, these have diversified over these 100 million years into all these activities, whereas the helpers, the executors, remain somewhat constrained and had limited expansion. Now, one criticism we had from the early days was, well, okay, you have a network, but big deal. I mean, every signaling pathway is a network. And a network, especially from the perspective of developmental biology, and in fact, it was a developmental biologist who made that criticism, where networks, they function into signal conversions, basically to compute a response, where you take in information coming from the environment, coming from multiple signals, and compute them into the appropriate response. So basically, network function into signal conversions, adaptation to the environment, amplification of the signal, etc. It's not necessarily that you have redundancy. But this is not the case. In our case, we have truly insulated pathways. These NRC pathways, even though they are working with the same sensors, they are actually insulated from each other. They are truly redundant. These are not partially redundant or anything like that. Like with RX, we could show very clearly that NRC2, 3, and 4 are equally able to confer full resistance to the virus independently of each other. So these are truly independently insulated pathways. And recently, we also developed biochemical evidence for this. For example, we can show that NRC2, even though it's redundant for some R genes with an RC3 and RC4, does not associate with an RC3 and an RC4. It only forms a complex with itself. So you can see here, this is an IP where an RC2 is IPing with itself, but it's not associating with an RC3 and it's not associated with an RC4. So these are truly biochemically separated pathways that are equally redundant. The reason I'm making a big deal about this is because this is important. Because why redundancy in biology, right? I mean, this is kind of a very basic question in biology. Why do we have redundancy? Why do we have two eyes? Why do we breathe with our nose and mouth? Of course, redundancy brings robustness. So redundancy basically means that, for example, if I get a cold and I cannot breathe with my nose and I can still breathe with my mouth, and I will survive, right? Otherwise, every time you have a cold, then you just die from suffocation, right? So the redundancy is very useful in biology because it creates robustness. It allows the system to be more resilient in terms of dealing with perturbations from the environment, etc. In our case, with plant immune system, how about evading immune suppression by pathogens? If you have redundant core elements, then you're more likely to evade suppression by pathogens. The second point is evolvability. And I'll get to this later because what I'll do now is I will tell you about why we think the network provides robustness. And then I will tell you about why we think the network also provides evolvability. And I think these two aspects, these two features really of networks explains why we have a redundant receptor network that has evolved in these immune systems. Okay, so let's talk about robustness. Let me check the time here. So, okay, good. All right, so very early in this project, when we discovered the network, we had a really powerful hypothesis, which we thought we should pursue. And then Lida Derevnina joined the lab at that time. She wrote a Marie Curie fellowship and she got funded. So she was very excited to focus on this. And I'm really glad that this project has now sort of reached fruition. And what she hypothesized is very simple. If we have this network in solanaceous plants, if we take solanaceous pathogens, bacteria, nematode, or mycetes, aphids, etc., we should find effectors that are probably going after these core elements, right? Because if you can suppress the NRCs, you're basically taking out of action maybe half of the NLRs, right? So it's a great target for a pathogen to go after and suppress, right? So what Lida did is she set up a screen, and this is now published. You can see it in bioRxiv, but I'll take you briefly through the story. She set up a screen with various solanaceous pathogens and pests, and did a screen where she looked for suppression of PRF and RPI BILB2. So these are two R genes, NRC sensors, that are NRC dependent on different NRCs. And to cut a long story short, she had 6 candidates that came out of that screen, 3 from the cyst nematode and 2 from Phytophthora infestans. And then she went on to test whether these are functioning at the NRC level or downstream, right? And that was a very easy trick to do that because we know how to introduce mutants into the NRCs to make them autoactive or autoimmune. So then they are active in the absence of a matching R genes or matching sensor. So what she found then is these 2 effectors, AVR-Cap1b from Phytophthora infestans and Sprycek15 from cyst nematode, potato cyst nematode. Both of these are really potent suppressors of an autoactive NRC. So this is an NRC3 that carries this mutation that makes it autoimmune or autoactive. On the other hand, they could not suppress NRC4. So there's specificity in this suppression, in this activity. They are suppressing NRC3 and NRC2, by the way, but not NRC4. So yeah, but that's the same data. And then we went on to study this, and the Sprycek15, so this nematode effector, turned out to be really interesting because this effector directly binds NRC2 and NRC3, but does not bind NRC4. So what you see here is co-IP experiments using the effector, the nematode effector with NRC2, NRC3, and NRC4. And if we look at the IP, then we see that we get a signal with an NRC2 and an NRC3 and a very hardly detectable interaction with an NRC4. And she went on to show that this is happening in the MBR domain. We had the independent results through a blind screen with hypergenics by Histo hybrid, and also the NRCs came out of that. We went on to do some in vitro work, confirm these results through work primarily by Abbas Makboul, showing that we can form a complex between the NBR domain of an NRC and this Sprycek effector. This is an in vitro complex. We even obtain crystals with the hope of getting the structure. But the reason I'm telling you this actually is a bit disappointing, because we did have these crystals, and we generated 3 rounds of these crystals, and they never diffracted well enough to get the structure. So, so I just want to share this with you, because this is actually frustration, many years of work, and we still basically have no data to show for. We know that these 2 proteins form a complex, but structural biology is like an all or nothing type of research. And in this case, despite all this work, we still don't have a structure. So we're still at the drawing board. But we are excited about the fact that we have an effect that directly binds an NRC, seems to directly act on it, presumably as an inhibitor. We still don't understand the mechanism. We were hoping the structure would give us a hint. But at least this is really interesting, the prediction that we made, that we have a core node in the network, and that we expect that an effector will target it. And we actually did find even an effector that directly binds that core node in the network. The summary, and again, I don't want to sort of tell you too much. The paper is out there, but I think the key point here is the take-home message. The take-home message is whatever works, right? So pathogens have evolved to target the NRC network at multiple levels. We have this SPRISEC15, which we know binds both the inactive and activated forms of the NRC resistosome, presumably. We also found this Phytophthora effector AVI-Capon-B, which we know is indirectly suppressing the NRC response by binding this protein Tol9, which is involved in endosome or vesicle trafficking. We don't understand, again, here the mechanism, but we're still studying that. But we know that Tol9 is important for the suppression activity of AVI-Capon-B. So maybe this is acting downstream of these activated resistosomes. And also we find other effectors that we didn't have time to study that seem to act at the level of RPI, BB2, and RC4 interaction, not downstream, and they seem to be suppressing this activity. But again, maybe the conclusion here, and going back to my point about redundancy, is perhaps one reason why these NRC have evolved to become redundant in plants was to evade pathogen effectors. So for example, let's take Solanum demissum, the native host of Phytophthora infestans out in Mexico. Some of you know that, but Phytophthora infestans is obviously a global pathogen in agricultural systems, but in its native environment, it actually originates from central Mexico and infects wild Solanum species like Solanum demissum. Solanum demissum is a hexaploid, and it has a very large family of NRCs. If you take these NRC2, 3, and 4 and look at all the paralogs and the 6 haplotypes, you'll find dozens of these NRCs, right? So our suspicion, and we haven't had time to actually get into this project, I'd love to do that one point, is that essentially the coevolution between effectors targeting these NRCs and NRCs evading the suppression is driving the emergence of multiple NRCs to evade suppression and keep interacting with these different sensor NLRs. So that would at least be one aspect of why we have a network in this NLR system. Okay, so now to finish, we'll talk about the second part, evolvability. So what is evolvability? So evolvability is, again, a really important concept in biology whenever we talk about networks, biological networks. And evolvability is essentially when you have a network, the system can be much more efficient and has a lot more room essentially to vary. I'm going to give you a metaphor because I love metaphors, and I think metaphors are always good to illustrate the point, right? Remember the penguin, right? So think about a restaurant. Imagine you have a restaurant and you have only one person running that restaurant. To be honest, I've seen places like that. You know, you get into some small village in Italy and there will be a restaurant and the guy would be, you know, cooking, hosting you at the table, getting you the check, doing everything, right? So that guy has really hard time managing a lot of people in that restaurant. That would not be a very efficient system. But if you have multiple players and they specialize in different activities, one person is cooking at the kitchen, one person is waiting at the table, one person is at the cashier checking, getting you the bill, etc. that system becomes much more powerful, much more evolvable, if you like, and that can accommodate now a lot more diversity. You can have a bigger restaurant, you can have more people coming to the restaurant, etc. Right? So it's a little bit the same. If you have one receptor doing everything, it's going to be constrained into what it can actually evolve into. For example, acquiring a new domain is going to be really hard to acquire a new domain and at the same time maintain the capacities to signal and cause the cell death through the traditional sort of classical pathway. So bottom line here is if we uncouple recognition from signaling into different proteins that are interacting together as a network, that system becomes more evolvable. There's a lot more room for that system to diversify and evolve. And this is what we think has happened. We started with multifunctional ancestors, and these have evolved into specialized receptors and specialized executors. But what about the data? What about the data? And also, can we look now at the diversity of NLRs we have, interpret them from this evolutionary framework, and ask the question, what are the molecular consequences of transitioning from a singleton into paired and networked NLRs that are specialized? Can we see biochemical evidence for this transition? And what are those biochemical signatures of this subfunctionalization? So let's go back to ZAR1. Remember I was telling you about the ZAR1 resistosome and how when ZAR1 is activated, there's this really beautiful flip of the N-terminal alpha helix, which is normally buried. And this flips out. Let's call it the death switch, if you like. I like to call it that because that's really what it is. And this conformational change results in this funnel formed by 5 of these alpha helices, 5 different proteins coming together. And these form this pore channel that is perturbing the membrane and functioning as a calcium or anion channel. So when the paper came out, we were actually very excited. That was one of the most exciting days of our lives because we had some data in the lab suggesting that the N-terminal of the NRC is really important for their activities. So of course, the first thing we did was to align our NRCs to ZAR1. And guess what? It turns out that the N-terminus of the NRCs is actually quite conserved compared to ZAR1. You can actually see here that the N-terminal alpha helix is roughly 50% identical between ZAR1 and NRC4. So that was really exciting. We had also independent evidence suggesting that in the NRCs, this N-terminus is really important for the cell death activity. So again, this is published from a couple of years ago, but to cut a long story short, this N-terminal alpha helix turned out to match this motif, which we call the MADA motif, or this MADA signature in the consensus of these proteins. And these MADA type CCNLRs are about 1/5, 20% roughly of the CCNLRs. And they're found in CCNLRs across all plant species, not just like Arabidopsis, Asterid, Solanaceae, but also like, for example, PIC2 is a rice protein. MLA is another one that has the famous monocot barley and other monocot CCNLR that also has a MADA sequence at the N-terminus. So this N-terminus is conserved in many NLRs, not all of them, 1/5 of them, but many of them. And also we could demonstrate in this paper back to 2 years ago that this is not just sequence conservation, but this is functional conservation. So we can actually take the N-terminus of multiple MADA type NLRs, including ZAR1 itself, and swap them into NRC4. So replace the NRC4 N-terminus with these N-termini of these various MADA type NLRs. And that NRC4 will still function and cause cell death, and even can cause resistance to Phytophthora infestans by functioning with the RPI-BLB2R gene. So this basically shows that our ZAR1 NRC chimera, where the N-terminus, the alpha helix is replaced, of NRC4 is replaced by the ZAR1 alpha helix, confers resistance to P infestans, as you can clearly see here. So this is not only sequence conservation, but also functional conservation across a large swath of NLRs from many different species. Now, back to our NRC network. What was amazing there is that the MADA type sequence we can only find it in the NRC executors. But none, absolutely none, zero of all the sensors carried this MADA signature. None of them had it. In fact, there are two major classes of sensors. I didn't tell you that, but for example, RX belongs to these sensors, which have the classic architecture, but PRF, RPI, BILB2, MI, and so on, all these other R genes belong to this green clade of NRC-dependent sensors. And all of these guys, all the green guys, have actually N-terminal insertion before the CC domain, so before actually where the MADA is supposed to be present. And that N-terminal integration, which can be quite large, several hundred amino acids, is absolutely important for the detection of the pathogen effectors. There's actually some really nice work with SW5 coming from Nanjing University, indicating the importance of this N-terminal extension, and also with PRF, nice work there, also showing the importance of this N-terminal extension. So these guys, because they have that N-terminal extension, they cannot function through the resistosome type model. What happens is all these sensors have diversified. They lost the MADA sequence because they don't need it. They're functioning with the NRC helpers, which are the ones that are executing the cell death and functioning like ZAR1 into causing hypersensitivity through this pore formation at the membrane. So bottom line here, main conclusion is use it or lose it. Basically, the sensor NLRs don't need the MADA sequence anymore. So they have basically, the MADA sequence has degenerated over time. That's the model we have, that as these guys went from singleton into specialized pairs, the sensors, they don't need the MADA sequence, so it degenerated. And then also some of them acquired even N-terminal extensions. They acquired new domains, which definitely make them not able to function through typical ZAR1 type resistosome. On the other hand, the helpers, through all this evolutionary time, dating back to monocots, dicots, and what have you, they retained the MADA sequence. That's basically the business end of these proteins. And also that MADA sequence has retained its conserved position at the N-terminus, which is obviously really critical from the perspective of the resistosome model. So you see now the point here that as we have this evolutionary diversification, we also have this specialized roles, this subfunctionalization, which also comes with biochemical signatures, where this protein take very different, very distinct evolutionary path into basically functionally specializing into either the sensing activity or the helper activity. And that makes this network a much more evolvable system, if you like, to keep up with pathogens. These guys are now freed to co-evolve with pathogen effectors, acquiring new domain, acquiring new mutations. Maybe some of them, the CC domain now is baiting the effector itself. Doesn't matter, it can lose sequences. All it cares about is really capturing effectors and not really being constrained by the necessity to execute the cell death. Beautiful, isn't it? So back to the penguin. If you want to understand NLR diversity, if you want to understand how this protein function, you cannot ignore the evolutionary perspective. You simply cannot. You're not going to be able to have a grasp of what these proteins do if you focus on one protein and ignore that perspective. Ignore the fact that they form receptor network, and ignore the fact that many of their features are degenerated, if I may say, or have basically diversified way beyond their ancestral function. So if I have one take-home message from the whole talk, it's really about this, is I want you to really remember the penguin. Beyond a uniform view of NLR structure and function, these proteins are very diverse in plants, they are rapidly evolving, and they also have evolved very rapidly in terms of their activities and biochemical functions. And that's really the take-home message I want to give you. So yeah, so that's all I had, and I hope this opens the door to great discussion. A lot of people contributed to this project. So Shihan is the one who really started this work. And Shihan is now at Academia Sinica. He was a PhD student when I started this work, and we really literally stumbled onto the NRC network. And that basically, he was a very influential PhD student, because now, as you can see here, half of my lab works on these NLRs. Aki joined the project, and he's the one who discovered the MADA motif. And he's also now just started his lab at NARA, at the NARA Institute of Science and Technology in Japan. And both of these guys are working on NLR, so I look forward to collaborate with them and follow their progress. Lidal is the one who did the suppressor screen, and she'll be moving to Cambridge to the Crop Research Center in January to continue working on suppressors of NLRs. And then various other people have been involved. Mao has done a lot in very different aspects of NLR biology and is current PhD student. Yorgos is the guy behind RefPlant NLR, and Abbas is our biochemist structural biologist. And I didn't talk about BLAST today, that's for another day, but I should also thank many people in the team, Adeline, Joe, and also some new people that joined us in 2021. In particular, Pi has already quite a lot of influence on NLR work, and of course my colleagues at the Sainsbury Lab. So thank you, and I hope we can have a fun discussion.